In this presentation, we're going to talk about a special case of diffusion, which is called osmosis. It's the diffusion of water across a semi-permeable membrane, meaning a membrane that allows some materials, like water, to cross, and other materials, like solute, not to cross the membrane. So osmosis is simply diffusion of water. It's a passive process, which means that water is going to move from an area of high concentration of water to an area of low concentration of water. And the reason why we look at osmosis or the diffusion of water all by itself is because water is really important to life, so we talk about it separately. So there are a couple perspectives that you can use to look at osmosis. So osmosis is the diffusion of water from an area of high concentration of water to an area of low concentration of water. So moving from an area where we have lots of water to an area where we have less water. Another way that you can look at this is by solute. Water moves to wherever there is more solute in the solution. And osmosis takes place across a very special type of membrane called a semi-permeable membrane. And this is key to having osmosis occur. Semi-permeable membrane means that the membrane allows some materials to cross the membrane and other materials not to cross the membrane. So in this particular case, water can cross the membrane, but whatever is dissolved in the water cannot cross the membrane. So the direction of osmosis is determined by comparing total solute concentrations. So as our penguin is reminding us, a solute is what gets dissolved in a solvent to form a solution. So for example, salt water, the salt would be the solute, it's what got dissolved, and the water would be the solvent. So salt water would be the solution, solute would be salt, and the water would be the solvent. Or if you were making Kool-Aid, the Kool-Aid powder would be the solute, and the water would be the solvent. So when we are predicting which direction water will move during osmosis, we compare the solute concentrations of the two adjoining regions. And we use some descriptive words to describe the relative solute concentration, meaning how much stuff is in that solution. So hypertonic means more solute, less water. So hyper is a higher concentration of solute. Hypotonic means less solute is dissolved and there is more water. So hypo means less solute. Isotonic, the prefix iso means equal or the same. So this would be a condition where you have the same amount of solute concentration on both sides of the membrane. So if we take a look at this diagram below, we can see these yellow dots are meant to represent solute that cannot cross this semi-permeable membrane. And we can see that we have way more solute on this side of the membrane than we have on this side. So less solute, we use the term hypotonic, and over here we have more solute in comparison to the other side of the membrane, so we call this side hypertonic. So one of the things that you can use to remember what direction the water will move is water follows solute, meaning that water is going to always move towards where there is a higher solute concentration. So water is going to follow the solute. We have more solute on this side of the membrane, so therefore the net movement of water is going to be from the left to the right. So the water is going to move from an area of less solute to an area with more solute. And this makes sense and still follows our rule of water moving from an area of high concentration of water, because where we have less solute, we have more water, and to an area with lower water concentration. So when you have more solute, you have less water. And the whole purpose of the water moving across this membrane is to create equal concentrations of solution on either side of the membrane. And the reason water is moving is because the solute can't. So looking at this and how this applies to living organisms, we have to look at cells and the environment. So the environment that a cell exists in is key to its survival. So here, all of these cells in the top row represent animal cells, down below are plant cells. So if we put an animal cell or a plant cell into fresh water, the water surrounding the cell has lower solute concentration. So fresh water means there's absolutely no solute in that water. So therefore, we would say that the environment that the cell is in, so the area outside of the cell, would be hypotonic. In this particular case, where we have a balanced environment, meaning the same concentration of solute outside of the cell as inside of the cell, we would say that the conditions or the environment was isotonic. In this last case, which is salt water, we have a much higher solute concentration, meaning more stuff in the environment, dissolved in the environment, than we have dissolved within the cell. And we would say that the environment, meaning the area outside of the cell, is hypertonic. So managing water balance. So how does a cell manage water balance with the different environments? So in a freshwater environment, meaning an environment that is hypotonic, has lower solute concentration outside of the cell than inside of the cell is going to be hypertonic to that hypotonic environment. Remember, we're always comparing. So we're going to compare the environment to the inside of the cell. The problem with a cell being in a hypotonic environment, meaning an environment with less solute than inside of the cell, is that the cell is going to gain water. Water is going to move into the cell because the inside of the cell has a higher solute concentration, which can cause the cell to burst, or the fancy word for bursting is to lyse or to split. So an example of this is a little single-celled organism called a paramecium. Paramecium live in a freshwater environment, and the paramecium's inside is more concentrated than the environment outside, and so water is constantly moving into the paramecium. So the solution that the paramecium has is something called a contractile vacuole that quite literally pumps the water out of the cell as it starts to enter, and I'll show you what that looks like in just a moment. Plant cells, however, are protected from bursting because they have a cell wall. And the cell wall around them will actually resist the movement of too much water into the cell. And at this stage, we say the cell is turgid or full. This would be when your celery is at its crunchiest. Your vegetables are at its crunchiest when your plants are standing as tall as they can, because each of the cells are filled with the maximum amount of water that they can hold. So a plant cell in a hypotonic environment, no problems. The cell wall will protect it. However, an animal cell or a paramecium cell, which is a category called protista, that is an issue because water will rush into the cell, causing it to burst. So here's a picture of our paramecium. It's a little single-celled organism, and these little hairs along the outside are cilia. And right here, you can see the contractile vacuole, and here the vacuole is filling with water. And here you can see the vacuole has contracted, and what gives the energy for that vacuole to actually contract is ATP. So what I'm going to do is actually show you a video from YouTube where you can see the contractile vacuole in. So here's our little paramecium. You can actually see the cilia moving on the outside of it, which would allow it to move. And here's the contractile vacuole, and it's full of water. And as it fills with water, contractile vacuole here, what happens when it fills with water is it squeezes and tightens and pushes the water out. We'll see that again. So here is our contractile vacuole. Here's a close-up on it. So the contractile vacuole fills with water, and when the paramecium has taken on enough water, it contracts and squeezes the water back out. So if a cell is in salt water, the environment is actually going to be hypertonic to the inside of the cell, meaning that the environment surrounding the cell is going to be higher in solute concentration than the solute concentration inside of the cell. And the problem with this is that the cells lose water, shrivel up, and in some cases it can cause the cell to die. So an example are shellfish. Shellfish, like clams oysters, they can live in saltwater environments. And so what they do is they actually take up water. So they actually, instead of pumping water out like the paramecium, they actually pump water in. Or they have salt pumps that will actually get rid of the excess salt inside of them. Plant cells, again, are protected by a cell wall, so they do something called plasmolysis. That's how this word is pronounced, plasmolysis. And what actually happens is the cell wall will keep basically the shape of the cell, of the plant cell, but the inner membrane will shrivel up. And we're going to see this in lab a little bit later. And so this is a condition where we say that the plant is wilting. And the plant cell can recover from this if you add more fresh water to the plant. So the plant will survive, the animal cell will shrink and shrink and shrink and may eventually die. But what we hope for with our cells is that they're in a balanced solution, or what we call an isotonic environment, where we have the same solute concentration outside and inside of the cell. And when we're in an isotonic environment, there's no problem. There's no net movement of water into or out of the cell. And we say that the cell is at equilibrium, meaning we have the same amount of water flowing into the cell as we have flowing out of the cell. So the same amount of water flowing in both directions. And it keeps the cell volume stable, the way that we want it to be. An example of this are the IV fluids given at the hospital. So when you're given an IV bag, there's actually salt and other solutes dissolved in that IV bag. They're not actually giving you just plain fresh water. If they did give you plain fresh water into your veins, it would actually cause your blood cells to start to burst. They would take in too much water and they'd explode. So IVs actually have a balanced solution that is very similar to what's called the blood plasma. Blood plasma is the liquid component of your blood. So believe it or not, the red blood cells that you think make up entirely your blood are actually dissolved in or floating in a liquid component called the blood plasma. And IVs are the exact same solute concentration as blood plasma. So in an isotonic environment, animal cells are nice and happy. They have the perfect flow of water in and out. Plant cells, however, could be a little bit better. Plant cells prefer to be in a hypotonic environment. They'd like to have lots of water flowing in to make the cell as turgid or as stiff as possible. So osmosis is not the only thing that explains the movement of water into and out of cells. Discovered in 1991, or published, the findings were published in 1991, was the discovery of what are called aquaporons. So these gentlemen here, Peter Eggery and Roderick McKinnon, what they discovered was that there was more things moving water into and out of cells than just plain osmosis. And these are structures, protein channels called aquaporons that actually allow for a cell to rapidly move water into and out of cells to create balance within the cell. So real quick, we're going to do a check for understanding. So here we have a little baggie that's going to represent our cell, and then we have a beaker with a solution that represents materials outside of the cell. Inside of the cell, we have a mixture of 0.03 molar sucrose, type of sugar, and 0.02 molar glucose. So this is the solute inside of the cell, and these are the concentrations of the three sugars that are in the environment, the area surrounding. So what we need to do is find the total concentration inside of the cell, and we do that by adding those concentrations together. So the concentration of the fluid inside of the cell is a 0.05 molar solution. On the outside, it's 0.3. So we have a higher solute concentration inside of the cell than we do outside of the cell. So the cell, this baggie, compared to its environment is going to be more concentrated, so therefore we call it hypertonic. The beaker, however, has less solute concentration, so the beaker, the environment around it, so we call it hypotonic. So which way will our water move? Water always follows solute, so water is going to move to wherever we have the higher solute concentration, which means water is going to move out of the bag, or I'm sorry, is going to move out of the beaker and into the bag. So the water is going to move to wherever it's more concentrated, so the water is going to flow from the beaker into the baggie, causing the baggie to swell or get bigger. And that concludes our lecture on osmosis.